Biophysics and the seminar series and the legacy of Dr. Groom. Okay, as we move on to the next part of today's seminar session, I would like to share just a brief bit of my own perspectives on Dr. Ellis. It's okay, Chris, we're okay. My first interaction with Western University was through hating your football team. And annual slugfest that left me with multiple concussions and shoulder injuries and other problems I live with to this day. But in 1989, 1990, so 30 years ago, I started to realize that my football career was time limited. And I started realizing I really like cardiovascular science. But Guelph doesn't have a lot of microvascular people, which is what really started to interest me. Not a lot of cardiovascular people, or it did back in 1989. So I started looking around in the literature, and I saw these papers by this guy, C.G. Ellis. And so I just started reading them as an undergrad. But they're really interesting papers. In fact, the papers on the structure of microvascular networks and skeletal muscle, the papers on hemodynamics, papers on how these things may impact mass transport and exchange, oxygen transport and exchange. These were incredibly interesting to me. And as a mark of how interesting, they were the first papers I ever read to the end. I was a great reader of titles and abstracts and said, okay, got it, moving on. Chris's papers are the only ones. These are actually interesting. I kind of like these. I could maybe do this for a while. And so 30 years later, I still am playing with these similar ideas. And a lot of this really comes back to reading and enjoying Chris's papers in the late 80s. Even as early as about 7 or 8 years ago, when I was going back to visit my parents who still lived in Guelph, I stopped here at Western for a day or day and a half, something like that, because I knew Ellis is on the way. And if there's someone who's going to have insight into some of the network questions and hemodynamic questions that I was having, it was going to be Chris. So I wanted to stop and pick his brain for a few hours, and really had a wonderful visit with him then. So, yeah, it's been 30 years that I've been reading your work, Chris. And I remember you were described to me back then as, yeah, he's a talented young guy. He's going to have a good career. So that's what you were to some people in 1989. So there you go. Those are my initial perspectives on Chris, and they really impacted me for a huge part of my career. But I think it's important that when he's being introduced for his retirement lecture or his final lecture, whatever we want to call it, that one of his longest collaborators and colleagues really provide that perspective. So we're thrilled to have Dr. Peter Canham, the long-term chair of the department, leader of this department, to come up and provide his perspectives and his recollections of Dr. Ellis. Thank you.
How do we get the slides to go? What do I push to? Thank you, Jeff, for the very kind words. It's a real pleasure and an honor to be asked to talk about our longtime friend and esteemed colleague, Chris Ellis. This first slide, this is the beginning of the becoming retired, I guess. Last spring, he's receiving his citation from the Chancellor at convocation. Oh, wrong button.
Arrow down.
Arrow down? Okay.
Yeah, I think so.
Oh, I'm on the wrong computer. The previous speaker remarked about Christopher G. Ellis, C.G. Ellis, and we actually have another Ellis, C.J., so we'll get into that. This is our lovable, friendly, cheerful mentor to students, wonderful research collaborator. He started his university career at McMaster University and followed by a very protracted PhD at Northwestern with Tom Goldstick. And in 1979, came to biophysics. It soon became apparent that he was a perfect fit to the department, both in teaching and research. And he came with a postdoc with Alan Groom. He had a very brief overlap with Alan Burton. And it's interesting because they really couldn't have much of a scientific overlap, but he did play bridge at noon with Burton. And I think he probably picked up some very unconventional bidding habits of Alan Burton. Things like, oh, diamonds are a girl's best friend. Or sometimes, boy, a good time to do some heart research if he was trying to give some hint about what suited him well. Thus, launching the exceptional career in cardiovascular research for Chris Ellis. Now, the right computer. There's another Ellis, a C.J. Ellis. In fact, I didn't know about him until we got to the not-so-funny part. But I do play bridge or play golf with him now. He was in the anthropology department, and he had expert knowledge in native settlements, two peak periods of 3,000 and 7,000 years ago, and he had digs near Grand Bend. And so this was maybe a funny mix-up. They had wrong phone calls, credit card inquiries, salaries, and sabbatical. But the not-so-funny was when this C.J. Ellis was starting his sabbatical leave in July 1998, and his salary was stopped. He thought, well, maybe he knew about the C.G. Ellis, whom I remembered as Canadian General Electric to keep him straight. But his salary was stopped and he couldn't pay his mortgage, so he was thinking, well, maybe the other Ellis has a rich bank account and he can weather the storm for a couple of months. But anyway, it was our Barb Anderson, our AO at the time, who actually solved the problem because the president's office was actually not very friendly and not very helpful in making this work out. Let's get back to our... He served as grad chair while I was chair for years and years and years. And he showed leadership, the program expanded, there was innovation. In fact, he earned the opportunity of being and served as Associate Dean Graduate Studies at Schulich for the graduate program. And he exercised various great things. He actually was a PhD examiner for a thesis in Holland. And when he went there, he was so impressed by the formality of the occasion, where everyone wore academic regalia, marching from building to building, that he brought back part of that tradition by having our public lectures for students in biophysics. They were going to be introduced by Chris in full academic regalia. And some grad chairs did continue with that, but I gather it's petered out recently. He helped us, well, I'll mention this too, important. He attended all the PhD defenses, which is really something because he learned how students were coping with answering questions and what good examiners we had and didn't have, and so on. And that stopped, though, when Graduate Studies said you can't attend unless you have special permission for every single lecture or every single defense that exists. So that did peter out. He problem solved for us, which was really helpful because Graduate Studies had left off the contributions of the teaching role in our graduate program. We had a big graduate program, and so that was a big loss, and that got sort of Chris's problem-solving skills. Chris can be a magician. He can sometimes disappear, and his lab certainly knows this. Yeah. And on one occasion particularly that I remember, the department office didn't know where he was, and his lab group didn't know where he was, and not even his cat knew where he was. He had disappeared to a posh hotel near Stratford to work on his grant renewals. Very nice. But actually, he's been very successful over the years with funding from NSERC, CIHR, or DCF, and all sorts of things. So it's a good strategy for those of you who would like to disappear to a Stratford posh hotel. I want to give credit, and there's a nice picture with Stefan Milkovich and so on in the Burton labs. He's probably on his third or fourth generation of this special microscopical equipment, but I want to pay special tribute to his technical skills. He's gifted and skilled with various technologies, and I think coming from chemical engineering was one of the best backgrounds you can have. But he received the first Microcirculatory Society Award for Innovation Instrumentation. And he received that for developing the first method for measuring oxygen saturation in single red blood cells in capillaries in a living animal. And that principle, we've actually had that presented in our first graduate student presentation today, was the changing wavelength absorption spectra associated with oxygen saturation. And he could do that quantitatively, which was a very fine way of making measurements. But not everyone appreciated Chris's talent. I was just by chance walking alongside Nigel Patterson, who's trained in internal medicine, very much a part of the clinical group at Victoria Hospital relating to microcirculation. And he just, out of the blue, said Chris's expertise is very fine, but he thinks it's just a technology looking for a place of application. I was dumbfounded, and I hope in my next slide that we can appreciate the incredible career that Chris has had and would argue against Nigel Patterson's concern. There's a picture with Alfred J. Alfred actually himself is a PhD graduate of Dr. Burton and has contributed largely to our graduate program through scholarships and prizes. So a wonderful person to help out our department. But when we look at clinical relevance, we know from the basic science that with UWO colleagues and many others, Ellis has studied the regulation of oxygen supply to tissues. Now, with critical care physicians, Ellis and others are investigating the clinical problems of microcirc injury and multiple organ failure. Now, Burton would have loved to see this career unfold because he actually held in disdain and disappointment the early microcirculation research with Zweifach and others, describing it as very descriptive and really lacking quantitation. And Chris has really shown how you can put numbers and concepts to work here. Much more than, of course, appreciating the rather, which Zoot did at Lunchtime Ridge. Please join me in welcoming Chris for the lecture that he's going to present. And thank you very much.
Well, this is a great pleasure for me to be here and speak to you. I want to really thank the department for this opportunity. And I need to correct a couple things that what Peter said. The other Chris Ellis, actually $1,000 came out of my paycheck. When they got a mix-up about the sabbatical, they took $1,000 out of my paycheck. I didn't know about it stopping on his, but Barb Anderson took care of that. That's the only one I want to correct. We'll think about others later. So I would spend some time trying to figure out what I would talk about. So how biophysics shaped my career seemed to be a good title. I mean, I've got 40 years of research experience here, and I've got 40 minutes to go through it. And so, and an academic career is a whole lot more than just the research that you do. It's all the courses that you teach, the administration you do, and it's the interaction with undergraduate, graduate students, and summer students. So there's a lot that's involved. So I tried to just pick out one particular thread, which was how biophysics shaped my career. And the first is, Alan Groom, he was my mentor. I sort of feel like some of the people who were talking earlier, like Jerry, actually must have seen my slides. So, and also Peter. So we'll talk a little bit about Alan, just briefly. A lot of us think about biophysics as the application of physics to study biology, the tools that we use and how we develop those tools. And I'll talk a little bit about how I've used biophysics to develop tools that I use to study the microvasculature. But the part that I really learned from Alan and really has influenced what I do is how physics defines biology. Because a lot of our biology is really determined by the physics that governs things. And so that gives me insights and understanding when I start to study the microvasculature. So let's go to Alan Groom first. I first heard him speak. I didn't know him as early as Jerry did. This would probably be the early 1970s or mid-1970s. I was at my supervisor had sent us all to a conference on oxygen transport to tissues, international conference. There was a lot of people there from Europe, from the Max Planck Institute, mathematical modelers, experimentalists, and I was there as a PhD student. I was very shy. I didn't really talk very much. When I saw Alan Groom, I never actually spoke to him. But there was this lecture one afternoon, and this gentleman stood up, a short fellow, white hair, English accent from the University of Western Ontario, and he started to talk about the microvasculature. And let me remind you, at that time we had physical slides. We didn't have PowerPoint. It was physical film slide. It was diazo, blue with a white lettering. There was no animation, there was no video. But he made the microcirculation come alive. And one of the things he did, let's see if I can find this, I had to borrow it from Jerry. He would reach into his pocket and he would say, you know, the mesentery is a tissue that a lot of microvascular researchers study. It's this very thin connective tissue around the gut. And it's so thin that you can really see the blood vessels and red cells going through them. In the body, it's sort of like this handkerchief. It's all scrunched up. But when we go to study it, we have to take it out and smooth it out, and we end up with this nice, beautiful, thin tissue that we can look at with a microscope. He was reminding us that whenever we go to study the microvasculature, we have to expose it in some way, and keep that in mind as you're studying it, especially if you're a mathematician who's developing a model that's trying to describe oxygen transport in that situation. I ended up a few years, I never did talk to Alan at that meeting. I did send him a letter later asking to be a postdoc with him, and he took the risk on me to actually hire me as a postdoc. I didn't have any papers for my PhD. My supervisor really didn't stress the idea of publications. I had papers from collaborations with others, but not for my PhD. And Alan took that risk on me. So this is where I start the research side of the story. What was the big research question at that conference? And what was it that ultimately led Alan to accept me? And it was really, what were the implications of microvascular blood flow heterogeneity on tissue oxygenation? And I think this is part of what Jeff is talking about earlier, some of these papers that I looked at. Implications of microvascular blood flow heterogeneity on tissue oxygenation. This is what the microvasculature in rat EDL muscle looks like. 10 power image, muscle fibers running in this direction. For some of you, you've seen this many times before because I just love this particular one. I'd like to show it. These little black dots, those are red blood cells as they go through capillaries. And as you look at it, you say, wow, here's an arteriole coming in, delivering red cells into the capillary network. It's draining out here in a venule at the top. But the flow isn't uniform. A lot of the people at the time at that conference, they really thought there was just uniform blood flow in all the capillaries, and that when you had low metabolic requirements, a number of these capillaries would not be flowing, and when metabolism increased, you would have a recruitment, an opening up of capillaries to deliver more oxygen. But in reality, this is what the situation looked like. And August Crowe, back in the 1920s, had won the Nobel Prize for developing a model and talking about how this capillary is supplying a cylinder of tissue around it. This would be really well supplied with oxygen, and all this tissue would be quite fine. But this tissue, the supply rate of red cells are much less delivery of oxygen. Maybe it would run out of oxygen by the end. And this was the concern that all of these mathematicians and experimentalists had at the time, is when you look at this, how come it's like this? Shouldn't it be much more uniform? How could you have a situation like this in terms of being able to maintain a uniform tissue oxygenation in an animal at rest in skeletal muscle? So my hook to Alan was, I want to study tissue oxygenation in a known microvascular environment. I would be really the first person to test Crowe's model in vivo. And many of the concepts that Crowe had, or many concepts that we think about, are derived from Crowe's work. So capillary recruitment, capillary reserve, critical role of capillary transit time on tissue oxygenation, all these things derive from Crowe's model. Is Crowe's work correct? So my proposed approach that I talked with Alan about when I came up to see him in 1978 was that he had a technique for looking at the microvasculature. Here we are looking at red cells going through capillaries. He could measure with Carl Timmel. Carl developed a technique for measuring the velocity of the red cells. I could go in and make PO2 measurements with a PO2 sensor in the tissue and know what the PO2 was in the area around those different capillaries with different levels of flow. And I would have flowing solution over the top, and I'd be able to stick my electrode in and make my PO2 measurements. I had the PO2 sensor expertise, and Alan and Carl had the expertise in measuring red cell velocity. Only problem, there's diffusion of oxygen out of the air into the flowing solution. That oxygen would then diffuse into the capillaries and the surrounding tissue. So the setup wouldn't work because it would be altering oxygen levels in the tissue itself. Oh, the other thing is that oxygen also affects blood flow. So I would actually be altering the blood flow because I was exposing it to different levels of oxygen. And so the blood flow would be changing at the same time. I had a situation where I could make all these measurements, but they wouldn't mean anything physiologically. So what do you do if your plan fails? Well, you do what you can. You've got to keep research funding coming in, right? So you've got to find some way to keep on going and doing what you can. So I took Carl Timmel's code for measuring velocity. I added in a bit about being able to do flow reversals, which was interesting, that blood flow would actually reverse in capillaries sometimes. And I measured how many red cells were in a capillary and how fast the red cell supply was. Not the velocity, but how many red cells per second are going through the capillary. Because that really tells me key hemodynamic parameters for the potential for delivering oxygen. And then I went ahead and I quantified the spatial and temporal heterogeneity in the frog sartorius muscle. I was an engineer working on rats at the time, or mice. That was a little bit beyond me surgically, so I worked on frogs, which is precisely what Alan was doing at the time. Here's the network that we looked at. I could measure all the capillaries in this field of view supplied by a single arteriole draining into a single venule. I could make measurements of all the velocities, lineal densities, supply rates in these vessels, and look to see what type of heterogeneity was actually there. So if I looked at velocity over 10 minutes in all these different capillary segments, I see a lot of variability from one capillary to another, and there's quite a bit of variability with time there as well. If I looked at the lineal density, how many red cells, or sort of measure of hematocrit in the vessels, there's even greater variability between the capillaries in terms of their lineal density. And you saw that in the video earlier too. And then if you take the product of these two, velocity times lineal density, you get supply rate. This is the potential for delivering oxygen. This capillary has got tremendous potential to deliver oxygen to tissue. This one's got very little. It really suggested that there's a problem with the idea of Crow, that each capillary is solely responsible for delivering oxygen to the surrounding cellular tissue. So, spatial heterogeneity of all these parameters that are critical for oxygen transport were persistent. Some people thought it just switched back and forth. If you got low flow for a while, it would increase, and you'd have high flow, and it would offset it. But I showed that it was a persistent, despite substantial temporal heterogeneity that was there. And this is one of the messages for the grad students. I could have stopped and published this data here at this point and just said, look at persistent heterogeneity. But there's a big data set, and I've spent a long time collecting that data and doing the analysis. Is there more that I can learn from it? And so I noticed that the supply of red cells coming from the arteriole into the capillary bed was highly variable. It would go up and down and up and down. And I wondered, does this affect the temporal heterogeneity that I'm seeing here? So does that variability in the arteriole affect what's going on in the capillary bed? And when I took the relative supply in each capillary relative to what was being delivered by the arteriole, you can sort of see that it starts to smooth out a lot of the temporal variability that's there. But I went a step further and I said, if I know this variation in these parameters over a time interval, and I divide by the mean value for that time interval, I get the coefficient of variation, that's a measure of heterogeneity. As my arterial supply rate goes up, my heterogeneity goes down in velocity, lineal density, and supply rate. Or conversely, with AJ is talking about preterm infants, if they've got very low blood flow, what would be happening with a low blood flow as the flow goes down? You would have an increased heterogeneity of perfusion in that capillary bed, making it more difficult to deliver oxygen into the tissue. So we've also observed the same type of thing in rats. So this isn't just something from frog. This is something that's essentially universal in a microvascular bed. So it's an alternative to Crow's capillary recruitment idea. Rather than active opening of new capillaries or capillary flow paths to reduce diffusion distances in response to an increased oxygen demand, we'll reduce the heterogeneity of the red cell supply to more uniformly distribute oxygen to the muscle. And that's achieved by passive rheology. It's just the properties of the way red cells travel through capillary networks. And I think Peter Canham would know something about that as well. And so would Alan. So it's going from active to something that's very passive and just a property. Now, I really want to get back and start measuring oxygen. Was there another way to do it than with the PO2 sensors? So I had two good colleagues that I've worked with, Roland Pittman in Virginia, and at the time Mary Ellsworth was a postdoc with him. They were attempting to use spectroscopy to measure hemoglobin oxygen saturation of red cells and capillaries. Back then, they had a video camera, they had an analog device to put a crosshair on the video, and they were getting chart recordings of the oscillations of light as red cells went by. And they were using a ruler and making some measurements at one wavelength of light, and then going on and doing it with another wavelength of light. It was really crude. So Roland suggested that we should collaborate, which we did. So AJ presented the spectral properties of hemoglobin. He's working way out here in this part of the spectrum, way beyond where I'm looking. I'm studying down in this area in the blue, purple and blue region of the spectrum, where a single red cell has a very high absorption of light. And so this red cell in this region will be almost black because of the high absorption of light at that wavelength. There's also a large change in absorption depending on whether it's oxygenated or not. So I can quantify, even with old techniques, the changes in how much light's being absorbed. Whereas over here in NIRS, the red cells will be essentially transparent. You have to have a lot of red cells before you can see them. Shine a very bright light source through the tissue, and through the muscle, and then through the capillary, and I measure how much light's absorbed at two different wavelengths of light, one which is a isosbestic, and the other is oxygen dependent. And I'm going to use Beer's law to be able to determine what the oxygen saturation is. But one of the tricks with all of this was, how do I measure the incident light, this incident light, because it's being hidden by the red cell. Nice thing about capillaries, red cells move. They move away, and I can measure the incident light when there's just plasma there. So I can measure the incident light and the light when the red cells are present in the capillary. I can put that rat on the stage of a microscope. By this time I was working with people who actually could do the surgery. It wasn't me, and I think I saw Steph in the audience a little bit ago. So doing all these experiments where the rat's on the stage of the microscope, the muscle is on a glass cover slip, isolated from the environment, so that it's not just to keep it from losing moisture, but to keep the surrounding oxygen from getting into the tissue. And now I can look at multiple capillaries and compare, instead of just a single PO2 sensor making a few point measurements, I can look at multiple capillaries in that field of view. And the technique involves light going down through a beam splitter to two separate cameras. I think we're probably the 5th or 6th generation now of technology that we're working with. Here's a video on the right showing what the video would look like, and then we process it to generate an image of where all the capillaries are that are carrying flow. We select a particular vessel that we want to do analysis on. We pull out what's called a space-time image that shows the movement of red cells through the capillary over time. Each one of these lines represents 1 or 2 red cells as they're going through, and I've color-coded it for oxygen saturation. So you can see when it's going slow, there's low saturation. As it speeds up, there's much higher saturation. So that's how the technology was developed and we use today. I can, from that, with the software that we've developed, measure geometry, hemodynamics of the red cells, their oxygen saturation. We get capillary surface area, oxygen flow rates, oxygen flux, a whole range of parameters, and this is all coming from just 2 sets of video images. And then we can take it a step further, and this is work with Graham Fraser and Dan Goldman. We can look at multiple capillaries in field of view and at multiple depths into the tissue, and from that reconstruct a 3-dimensional network of vessels. And that was the work that Graham did, and then Dan, with his modeling, could predict what the oxygen levels would be in the tissue. And this was the very first time that anyone had developed a way to use real in vivo data to generate what the tissue PO2 would look like around a capillary bed. So this is the capillaries. We know all the velocities and supply rates and saturations. And then this is the computed from the model what the oxygen levels would look like in the tissue surrounding it. One thing to notice is that despite a spatial heterogeneity in all those parameters of velocity, lineal density, and supply rate, the tissue PO2 is relatively uniform everywhere. If it were Crow, we should see a whole series of concentric circles around each capillary with high oxygen at the capillary falling off into the surrounding tissue. We don't see that. All of these capillaries in this volume of tissue are working together to deliver oxygen to the tissue around it. And that was a great insight to have. Finally, in 2012, Finally, what I said I was going to do when I first saw Alan, I'm able to do it. Tissue oxygenation in a known microvascular environment. It took a while to get there. It was the first computational model based on true in vivo data in the same animal, in the same microvascular bed that had been done. What about August Crowe? Well, August Crowe was correct. Diffusion places a really significant limitation on tissue oxygenation. That's physics determining biology. But a capillary isn't the sole source or solely responsible for oxygen to the cylinder of tissue around it. You can have diffusion from other capillaries to support those that don't have enough, at least at low and modest oxygen consumption rates. There's no need or evidence for capillary recruitment or all the other concepts that people have about when you have a stimulus, there's actually an increase in capillary volume. What about the implications for diffusional exchange? Capillaries, passive rheology, diffusional exchange, all ensure that there's going to be an adequate tissue oxygenation as long as you deliver sufficient oxygen to the volume of tissue. But what volume of tissue? It's not just that little cylinder of tissue. It's what volume are we talking about? Is it the volume of tissue supplied by that capillary unit? Or is it muscle? What actually is it? Another paper by Groom. They looked at capillary networks, published this back in 1976. It's about the time, just a little bit after I first heard him speak. And he took all of that data and developed a really nice network model in terms of all the parameters associated with it, and learned a great deal from it. Here's the one that I used, the frog sartorius muscle, just a single capillary unit. Here's the way I think or thought of how oxygen was regulated. Here's a single capillary unit supplied by an arteriole, and we would make sure that each capillary unit gets its own specific delivery of oxygen to match whatever the tissue needs were around it. And I'm not the only one who thinks that. There's many others that still are publishing, looking at single capillary units supplied by arteriole and venule. But arterioles deliver capillaries, deliver blood flow not just to a single capillary unit. They always deliver to at least 2 capillary units. If you take a look, you can see here going to a group of capillaries that are going up the stream, and here a group of capillaries going down the stream. Here it is. This is from Asher Mendelssohn's work. It's a montage of 3 different fields of view. And if you take a close look, here's, let me see. Here's where the arterioles are coming in. Here's where the venules are going out. And if you follow this, initially it looks really very, very complex and very difficult to understand. But as you start to follow this through, see these arterioles are supplying this group of capillaries here. These arterioles supplying this venule here, but also sending off in other directions. But there almost appears to be an underlying structure to this, to the way that microvasculature is supplying this tissue. Asher went through and very carefully used some of our functional imaging techniques to generate an image of one of those areas of the tissue, and then carefully traced out all of the capillaries that would be there. And as we see, here's an arteriole coming in, branching into terminal arterioles that deliver into this very beautiful network of capillaries. And as we look at this, Asher coined the term a microvascular lattice to highlight that there's a long network of capillaries with interconnected arterioles and venules connecting into it along the length. And here is with the confocal microscopy, looking at a stain on the capillary endothelium. And again, you get the sense that there are these long columns of capillaries that are very carefully organized, and there also seems to be almost a space in between them, that they're very discrete. So if there's this column or bundle of capillaries, is there any corresponding functional tissue within the skeletal muscle that would correspond to these columns of capillaries? And I think Jeff Pickering is the one who said, "Do you mean a fascicle, Chris?" Which I'd never heard of before. But a fascicle, a muscle is made up of individual muscle fibers, but those muscle fibers are collected together into a fascicle, which is surrounded by a connective tissue sheath, and then there's a whole series of fascicles that actually make up your muscle. And this is what makes the muscle as strong as it is. There will be a mixture of muscle fiber types within the fascicle, but along the length, they will essentially extend the whole length of that muscle. So we have this structure of muscle fibers, but within the fascicle, now we would propose that there's a capillary bed with random locations where terminal arterioles and venules will come in and supply the capillaries. So we propose that only the terminal arterioles and collecting venules penetrate into the fascicle, and all the higher order arterioles and venules occupying the interstitial space between them. And the capillary lattice forms a continuous network of connected vessels from one end to another. Well, here I've sort of put them together. You start to see this is going to go on and on in both directions. So we can't really just deliver oxygen into a single unit. When we're starting to regulate it, we're going to be regulating it into two separate units. And what happens up here or down here is also going to influence what's there. It's a totally different structure and framework for us to think about. They'll also, each of these individual bundles will have different resistances in terms of the way the flow will be distributed. So if the higher order arterioles are not connected into the fascicle, how do they know how much oxygen to deliver to make sure that that tissue gets the right amount of oxygen? It can only work if we have feedback that's regulating oxygen, the feedback from the capillary bed within the fascicle, within the lattice, feedback that's going back up to the arteriole to do that. Different people who've been in the department, Hans Dietrich, Carl Timmel, others in the United States or in Guelph, have already shown that there's conducted signaling from capillaries into arterioles. We would say that there is such a conducted signal, and we wanted to look to see whether we could actually measure that independent of any oxygen metabolism. Can we just look at whether the system can regulate oxygen delivery or not? I'm now going to be flying through ideas that have been generated over like 20 years, okay? So one of my grad students, Justin Jagger, showed that we can release ATP from a red blood cell based on the change in oxygen saturation within the red cell. As saturation goes down, red cells release ATP, binds to receptors on the vascular endothelium, causing a conducted signal that goes up the endothelium, causing vessels, arterioles, to either vasodilate or vasoconstrict, depending on what the level of that signal would be. That was when we first thought all of this occurred within the arteriolar tree, but now we recognize that it must also be going on within the capillary bed. And so here's a single red cell going through this capillary here. It's got a very small space to hand off its ATP across a small plasma junction between the red cell and the endothelium, causing a change in membrane potential, which then can be conducted up into the arterioles because all the endothelium are electrically connected via gap junctions. So how do we study this in vivo? Well, we put a gas exchange chamber underneath the muscle and a oxygen permeable area that we can alter the oxygen or perturb the oxygen level at the muscle surface. Here is when you're looking at, through the microscope, a recording of it, This is when the muscle is exposed to a little bit more than normal oxygen levels. We're going to drop it down to 15 millimeters of mercury. So it's going from a little bit above normal to not really hypoxia, but a low oxygen level. And you can see the real-time response to that change in oxygen levels. That's just over the physiological range of oxygen. You can see how the tissue responds. So if this is the model of the lattice that we have, there has to be conducted signaling coming from the lattice up through the terminal arterioles, up into the larger arterioles to be able to increase the amount of flow necessary to meet changes in metabolism. This is all dependent on electrically coupling via gap junctions, connexin 40 gap junctions. We needed to test whether or not connexin 40 knockout mice respond to local oxygen challenges or not. So putting a connexin 40 knockout mouse or a control mouse, we exposed it to baseline oxygen, to a challenge, and then back up again. Here's the change in that space-time image showing that there is a change in saturation. Here's our measurement of the decrease in saturation back up. So this is pretty much the saturation around 65% is a normal level in capillaries and skeletal muscle. We're not taking a depth really that far, and yet we see immediately there's a response in terms of velocity increasing. We return back to normal with a recovery period. We see that the oxygen saturations come up, velocities drop. Not much happened with hematocrit, but a little bit. And the supply rate responds dramatically to us extracting oxygen out of the tissue through our exchange chamber. This shows in a control situation that we can alter red cell supply rate by altering the oxygen levels at the surface of the tissue. If we then compare control animals to connexin 40, connexin 40 do not respond to the oxygen challenge, whereas the control animals do. Connexin 40 is necessary for O2 regulation to occur. And then Richard Sauvé developed a really neat way for us to look at very small region in the tissue. How small an area can we affect with oxygen and still get a flow response? So he took a very thin glass cover slip, made an ultra-thin gas exchange chamber. It's only 400 microns thick. This window that's cut out of it is 200 by 400 microns, so we only have a few capillaries that are exposed. We cover it with a highly permeable oxygen membrane, and we then change the oxygen levels in the chamber. We'll be changing oxygen level over these capillaries, but not in any of the ones around it. So we're only testing, can we change flow by changing the oxygen levels here. The next movie is going to show this field of view at 20 power, centered on this portion of the window, and we'll see what the response will be. PO2 in the chamber with a pretty well normal tissue PO2, and we got nice flow. We increase it to 52 millimeters of mercury, partial pressure in the chamber, and we pretty well shut down the flow. There's much less flow going through those capillaries. We're only affecting the oxygen in this area, and we can see the response. What happens if we reduce the oxygen level? What we see is flow is returned back to normal. So that's all within the same animal using this technology that Richard developed for us. So those are new tools that we'll be able to use to explore even further about the O2 regulatory system with new technology that we have. Summary of 40 years. At least this portion of the 40 years, okay? No need or evidence for capillary recruitment. If you read it in a textbook, you read it in a paper, or you see someone talking about it with the technology that they're using, just remember, I told you, there is no recruitment, okay? Capillaries, passive rheology, and diffusional exchange ensures adequate tissue oxygenation if there's sufficient oxygen being supplied to the volume of tissue. The microvascular lattice and the muscle fascicle provides a new framework for exploring and interpreting O2 regulation. One of the things I mentioned way early on is there were flow reversals that I had to adapt the software to deal with flow reversals. You only get a flow reversal if you've got something like a lattice because of the way the network is put together. The integrated conducted signaling from capillary bundles to alter arterial tone is necessary for a functional O2 regulatory system. You have to have conductive signaling. Red blood cells are the sensor of the O2 regulatory system by an oxygen saturation dependent ATP release. That's a strong hypothesis. I think we have evidence for it, but it's still one that needs to be really nailed down and demonstrated. What happens in disease to impair this system and the impact on tissue oxygenation is a story for another time. I just could not fit in everything about sepsis and a bit of work that we've done in diabetes into this particular talk. But you can start to imagine, as you look at the way this system is designed and the way it's regulating, there are various places where disease could impair either the red cell function or the conducted signaling or the plugging of individual vessels and how that would ultimately affect tissue oxygenation. Now, reflection. A single superb presentation from Alan Groom is why I'm here today. Okay? If I hadn't heard his talk, and if he wasn't such a good speaker, I would not have done all of this research. Biophysics is more than applying physics to study biology. Physics, in many ways, defines the biology. And take time to reflect on your research. You know, you get caught up in everything that you do. You're trying to make deadlines. You need to take time to just step back and think about it. That's something that I learned from Alan as well. Think about what you're doing, reflect on it, let it digest and work through. It's really very important. And don't be afraid to have fun along the way. Alan had a great sense of humor. He was always fun to work with and discuss ideas with. Play fosters creativity. Just keep that in mind. Now, there are so many people to acknowledge in terms of research. Different funding sources that we've had over the years. I just put a rough list of different researchers that I collaborated with or continue to collaborate with. My graduate students, my summer students, undergraduate students, a whole range of people that have really contributed to all of this. And I should also say, my wife Kathy, because she's had to put up with me and my disappearing to not a hotel, it was a B&B.
Airbnb.
Where they would feed me. I could stay up all night and work on my grant and they would feed me. Kathy has put up with a great deal over the years for me. She really thought, we're only coming to Western for 2 years, maybe 3 as a postdoc. But the environment that I found here, the collaborative environment that we treated all of our faculty, our students, whether they're undergraduates or graduate students, we treated them all the same. And that wonderful environment was something that I wanted to stay and develop my career around. So thank you very much for your attention and a great audience.
Thank you.